Well, good day, everyone. My name is Hanadie Yousef, and it's a pleasure to have the opportunity today to share with you a bit about Juvena Therapeutics, which is a regenerative medicine startup dedicated to developing protein-based therapeutics that can regenerate tissue to cure disease and rejuvenate lives. Specifically, our protein therapeutics are being developed to treat age-related and degenerative diseases, and we're using state-of-the-art proteomics and machine learning in order to select protein therapeutics that can enhance aged primary human precursor and stem cell function, promote better differentiation and survival, leading to reduced fibrosis and ultimately tissue regeneration. And we're doing this by unlocking the unlimited regenerative potential of a subfraction of proteins that are produced by human embryonic stem cells. Specifically, my colleagues and I, during my time as a graduate student at UC Berkeley, and then much more extensively at Juvena Therapeutics, have shown that by isolating and concentrating a subfraction of the proteins produced by human embryonic stem cells and adding them either directly onto, for example, aged human precursor cells, we could enhance their function. When we add them virtually to every tissue type and cell type we've tested so far, they have impressive regenerative capabilities that really target the degenerative mechanisms associated with aging. We are first focused on developing therapeutics for skeletal muscle diseases. So as an example, when we add this cocktail of proteins either onto aged primary human muscle precursors, in vitro, we can enhance their proliferative activity and ability and myogenic potential. When we inject these proteins into acute injured aged mouse muscle that has poor regenerative capacity at that age, we can really rejuvenate muscle regeneration to levels of younger healthy animals. When we add these proteins, for example, onto treat them primary aged human chondrocyte precursors isolated from either aged knees or osteoarthritic knees, we could enhance the chondrocytes' proliferative capacity as well as their survival. And when we add these proteins, for example, onto human cortical neurons that are dying in the face of A beta treatment, we could actually prevent that death and promote their survival, even in the presence of these amyloid plaques. And so it's our goal at Juvena to really translate these proteins into therapeutics that can target and treat and cure age-related and degenerative diseases. However, it's not feasible to take what could be hundreds of unknown proteins produced in very small quantities from embryonic stem cells and try to develop that into therapies. There would be massive regulatory hurdles. It could potentially be unsafe. And so in order to do this at Juvena, we have really identified exactly what the proteins in this regenerative cocktail are using unbiased quantitative mass spec-based proteomics, where we, for example, compare the abundance of the regenerative subfraction that has immense regenerative capacity from human embryonic stem cells compared to the same subfraction of proteins produced and secreted by differentiated cells that don't have regenerative capacity. And so by really identifying what these proteins are, it turns out that there's about 1,000 abundant bioactive signaling proteins in this cocktail. And the next step that we have taken is to really then establish an efficient method, a machine learning enhanced discovery and preclinical development platform in order to identify the key drivers of rejuvenation in this cocktail, as well as simple combinations that could mimic the original cocktail to then identify, screen them in high throughput screening assays using primary human aged and diseased precursor cells and in vitro disease models, followed by extensive preclinical validation in a given disease model. And then ultimately, with the identified rejuvenating proteins, engineering them and lead optimizing them into biologics that can be used to treat patients with novel IP protection. And just to share a little bit more about our growing machine learning toolkit that we use for this efficient discovery and high throughput screening, So as mentioned, we've developed a machine learning enhanced platform in order to identify and bring these therapeutics to market. And specifically, our deep learning-based models assist us in using quantitative structure analysis, the various properties of proteins, for example, the evolutionary parameters, the chemical properties, as well as the vast amounts of scientific literature to really understand and predict and rank order which proteins in our library can specifically enhance regeneration of a given tissue type and disease. We then test this through rapid in vitro screening, where we've developed, for example, a deep learning model that will use bright field images alone to predict fluorescent labels indicative of the regenerative state, as well as the relative age of the treated cell to assess the level of rejuvenation, and models that can help us accelerate the quantification of histology, for example, the level of regeneration and fibrotic index from tissue treated with our candidate protein therapeutics. We are first, with laser-like focused on developing a pipeline of biologics that can treat skeletal muscle disease. Skeletal muscle being one of the largest internal organs, highly sensitive and integrated to multiple other systems. And unfortunately, it's a fact of life that once we hit our 30s, we're losing muscle strength and mass, which accelerates every decade and leads to increased incidences of diseases such as sarcopenia, acute fall injuries, disuse atrophy, especially after post-surgical aftercare, muscle wasting due to ICU, and there are also genetic myopathies such as dystrophies that have immense muscle degeneration. We now have 3 lead candidates in our pipeline that, based on the specific mechanisms of action of these protein therapeutics and signaling cascades, have been tailored for specific skeletal muscle diseases. So as an example, I'm going to show a little bit of data on our top lead, JUV161, which we would like to translate first to the clinic for muscular dystrophies. And so as an example, JUV161, when added onto aged human muscle precursor cells, can dramatically enhance their ability to fuse and form fibers. And these are aged human precursor cells isolated from an elderly individual. When we add JUV161 injected into acutely injured aged mouse muscle, we can enhance regeneration, specifically, almost to levels of younger healthy animals. And when we inject JUV161 systemically into aged mice that have this a reduction clearly measured of muscle function, we could improve that strength and function. So, for example, we could enhance grip strength and performance on a treadmill, and these are key endpoints that we would use for clinical trials for most muscle diseases. So, for example, we try to measure in mice things that would be indicative of improvement in humans, the 6-minute walk test, short physical battery of tests, increased strength, ability to get out of a chair, walk up a flight of stairs. And also JUV161 can prevent atrophy in younger mice that are treated with dexamethasone, which is especially relevant today given dexamethasone is now a treatment that is used, for example, for COVID patients. Ultimately, at Juvena, we would like to be able to translate our regenerative protein therapeutics to protein-specific therapies that can treat a variety of degenerative diseases. So our library, through our machine learning enhanced JuveNet platform, we are focused on identifying leads that can then treat diseases like neurodegeneration and osteopathies in the future. I'll quickly mention that using natural protein therapeutics as a class of biologics for regenerative medicine may have significant advantages over other approaches. For example, cell transplantation, while it's been successful in the area of hematopoietic stem cells and bone marrow transplants to date, has not been very successful for other cell types. And in fact, often the effects that we see might be due to paracrine signaling, while the cells that are transplanted themselves often don't survive. Gene therapies, while are going to be, incredibly successful in the future, really currently are really good for autosomal recessive diseases, but aren't that great in treating either autosomal dominant or heterozygous, heterogeneous mutations, as well as the current viral vectors aren't great at transducing muscle and stem cells specifically. The team at Juvena is comprised of myself and my co-founder, Dr. Jeremy O'Connell, who's a proteomics expert, Steve Julesgaard, who is a former executive vice president of Genentech. We have an incredible team of R&D researchers from top institutions such as Stanford and UCSF, as well as a host of industry veteran advisors from top companies advising us on our preclinical and clinical strategy. And we have an amazing scientific advisory board, which includes Professors Irina Convoy and Dave Schaefer, who are my co-thesis advisors and co-inventors of the original patent where we discovered these rejuvenating proteins, as well as Professors Joe Wu and Peter Jackson, Stanford professors with expertise in regenerative medicine and protein biochemistry. With that, I'd like to end by saying that we are very excited to currently seek partnerships and funding in order to advance our lead candidates and preclinical programs to IND-enabling studies in clinical development, as well as expand our platform to additional targets in degenerative diseases. Thank you for listening. Apologize for the technical glitches, but hopefully you got the gist of my presentation, and I'd be happy to take questions.
Hello.
Thank you, Hanadie. Jeff?
Great. Let me just go back on screen. Excellent. Well, thank you for the presentation. So if you could just remind us again what you're looking to raise for this, and what kind of milestones you'll seek to achieve with the funds that you raise.
Sure. To date, we've raised $7 million. We are seeking a Series A in the range of about $20 million in order to take our lead candidate and lead program through IND-enabling studies and into phase 1, as well as be able to expand our platform to continue identifying leads for other exciting therapeutic indications.
Great, great. And so can you talk about some of the, so you'd go in with, I guess, JUV161 as the first indication, followed by 191 next. And are the clinical trials already designed? Do you have a protocol synopsis built for the first one at this point?
We're working on that now. We have done a very thorough indication selection analysis and chosen a lead program based on the ability of 161 to specifically treat, it's an autosomal dominant disease, myotonic dystrophy, and in the midst of really then designing that phase 1 clinical endpoints and trial.
And how many patients do you expect to enroll in that trial?
We're figuring that out right now.
Oh, great. Okay, good, good. Well, thank you for the presentation.